Welcome to the Development in Sea Urchins module. In this talk, we're going to learn about gastrulation in sea urchins. Sea urchins are marine spiny globular invertebrate deuterostomes that belong to the phylum Echinodermata. They have been used widely as an organism to study different developmental processes. Sea urchins show external fertilization, and the zygote that is formed will then undergo radial holoblastic cleavage to ultimately form a blastula. The blastula is a hollow ball of cells with a fluid-filled interior, which is the blastocele. The blastula will then hatch from its fertilization envelope, and gastrulation will begin, which will result in organogenesis happening that will ultimately give rise to a larva. The larva is also called as a pluteus larva and will undergo metamorphosis to form a juvenile sea urchin. The juvenile sea urchin will then grow to form an adult sea urchin, which will then attain sexual maturity and be able to form gametes.
Sea urchin gastrulation occurs after the blastula is formed. Now we saw before that the late blastula is basically like a hollow ball. It is made up of about 750 epithelial cells that are in the periphery. And so in the center, there are no cells there, and that is where we have the blastocele. Now once the blastula hatches from the fertilization envelope, the large micromere descendants undergo epithelial mesenchymal transition, which is abbreviated as EMT, and actually enter the blastocele. This kind of movement, just as a reminder, is ingression. That is what is basically being shown in the figure below. So if we look at the blue box, we're actually seeing cells that are undergoing epithelial mesenchymal transition. And this ingression causes changes in the shape that we can see in the second figure for the embryo. And in the third panel, we can see more and more cells undergoing EMT. These cells are the descendants of the large micromeres. Let us now see how epithelial mesenchymal transition occurs. So before we go into the process, let's just have a look at the figure that is shown below. So we're looking at a layer of epithelial cells. These are the cells that are at the periphery, and the blue box shows the part of the epithelial cells that are in contact with the blastocele. And so they're covered with an extracellular matrix, and then they also have a basal lamina. When we look at the other side, these cells are interacting with the hyaline layer, and they also have their celial appendages. Now, during the epithelial mesenchymal transition, what happens is the large micromere descendants that are going to undergo the EMT, they lose affinity for the neighboring cells and also for the hyaline layer, but develop a very strong affinity for the basal lamina and the extracellular matrix components that are the parts that are facing the blastocele. There are many steps that happen that are involved in the EMT, and we're going to look at each one of these steps. So the first one is the apical basal polarity. Now the vegetal cells, which are shown right at the bottom in the black and white image, they elongate to form a thick vegetal plate epithelium. And that is what we can see in the black and white image, where at the bottom we can see that the ring of cells are a lot thicker than the ring of cells that are there on the top. The next step that happens is there's an apical construction of the micromeres. So the micromeres, the large micromere descendants that are going to undergo EMT, they change their shape and become constricted at the apical end. So when we look at the figure, if you focus on the portion that's in the blue box, we can see that the cell has changed its shape and it has become more narrow or constricted at the apical end. And the apical end is the part that is facing the hyaline layer, or that is away from the blastocele. There is basement membrane remodeling, so the micromeal cells will secrete enzymes like proteases to digest various components of the basement membrane so that they can migrate more effectively. The next step involved is deadhesion. In the process of deadhesion, the membrane proteins cadherins that we know play an important role in cell-cell connections, they are downregulated and degraded, and hence the cells are able to become free of their neighbors. And that is what is represented in the small blue box in the figure. Finally, we have cell motility. The cells, now that they have detached from their neighbors, can actively migrate out of the epithelium and into the blastocele. EMT is a very important step, and here in the figure where we can see that blue box, we actually can see a cell that is free of all attachments with its neighbors and can now migrate. Now, the migrating skeletogenic mesenchyme cells interact with the ECM proteins after the epithelial mesenchymal transition. Let's just look at the figure that is shown here. And when we look at the upper panel, we're actually looking at the embryo that has been stained with laminin protein, which we can see in pink, that is forming the inner ring, and the blue is the nuclei that is there of the cells. So right underneath the nuclei, we are seeing the laminin protein. When we look at the second or the middle figure, we can see that during the process of EMT, the laminin, which is an extracellular matrix protein, is being degraded. The green cells that are shown are the cells that are the skeletogenic mesenchyme cells, and we can see that they are undergoing ingression. And finally, in the last figure, or the one to the extreme right, is where we can see more and more mesenchymal cells undergoing ingression. When we look at the figures that are below them, we're actually seeing a cross-section of the embryo. And when we look at what is in the blue box, we can actually see these circular structures, which are the skeletogenic mesenchyme cells that have undergone ingression. And the hair-like structures that are right above it, throughout in that big hollow, are the extracellular matrix proteins. And so when we look at a more magnified version of this, we can actually observe that the skeletogenic mesenchyme cells interact with the ECM proteins, and they actually use those proteins to enable them for migration. Now, how do these cells actually know where they're supposed to be? So, there are two paracrine factors that play an important role in positioning these skeletogenic mesenchyme cells, and they are vascular epidermal growth factor, or VEGF, and fibroblast growth factor. Both of these are made by some ectoderm and endoderm cells, and they help in positioning the skeletogenic mesenchyme cells so they're in the right place in the developing embryo. The picture that is shown right below shows the staining patterns of fibroblast growth factor, and we can actually see that the fibroblast growth factor is made in only certain parts of the embryo, and they're shown in dark blue. And when we look at the lower panel of that figure, the blue cells are the cells that have the fibroblast growth factor receptor. And so the skeletogenic mesenchyme cells that are undergoing ingression actually have the fibroblast growth factor so that they can identify and bind to FGF and go to their appropriate positions. Now, when the skeletogenic mesenchyme cells leave the vegetal region, the remaining ventral plate cells will occupy that space. Now, during the process, the vegetal plate cells will undergo invagination into the blastocele to form a structure that is called as the archenteron. The archenteron is also called as a primitive gut, and the archenteron is basically going to give rise to the digestive system. So when we look at the figure that is shown, in the blue box, that is where we are seeing the vegetal plate cells undergoing invagination, and they are going into the blastocele. Now, during this process of invagination, an opening is formed right at the bottom, and this opening is the blastopore. So when we look at the image and the blue box, we can see the archenteron, which is that elevated hill-like region, and the blastopore, which will be right at the base, which actually is a hole that is formed. The same process is actually shown in these black and white pictures where you can see the formation of the blastopore as well as the archenteron in the one that is at 11.5 hours. Now the archenteron elongates to form a tube structure, and this process happens to various cellular rearrangements. Now the endoderm cells proliferate, they slide past each other and finally intercalate to elongate as well as narrow down the tissue. So when we look at the figure, the one in the blue box, we can actually see that the archenteron has become longer than the portion that or figure to the left, and it has actually become even narrower. So if we look at the size of the blastopore is much narrower compared to the figure that is right to its left side. This process where cells will undergo intercalation to try to elongate and form a narrow structure is called convergent extension. Now, non-skeletogenic mesenchymal cells also play a role in the complete development of the archenteron. They are actually present at the tip of the archenteron. These non-skeletogenic mesenchymal cells will extend these appendages called filopodia that will contact the blastocele wall. That is what we are seeing in the figure that is in the blue box, where the pink cells are the non-skeletogenic mesenchymal cells, and the ones that are right on top of that archenteron, they are actually giving out these red thread-like structures, which are the filopodia, and they're contacting the blastocele wall. Now what happens is the cells that extend the filopodia, they will actually start contracting. Now the filopodia bind to junctions between the blastomeres and they shorten to pull the archenteron upwards. And that is what is shown in the figure in the blue box, where we can see that the length of the filopodia have become shorter and shorter, and in the process it is pulling the archenteron more and more upwards, and this causes the extension of the archenteron. The same process is shown in these black and white figures, and there we can actually see the filopodia when we look at the picture in the book, but I'm not sure if it's that clear here. But what we can definitely see is the extension of the archenteron. Now the archenteron is the digestive system, and when we think of a digestive system, we need to have two openings. One is the mouth, the other is the anus. So let us see how these are formed. Now the archenteron extends to meet the blastocele wall. The place where the archenteron meets the blastocele, those cells will start forming the mouth. Now the anus is formed where the blastopore was formed. So when we look at the figure, in this figure here, we can see the archenteron touching the wall of the blastocele. And where it is touching the wall of the blastocele, that is where the mouth will be formed for the larva. The opposite end, which is where the blastopore was formed, that is where the anus is formed. Since the anus forms where the blastopore is formed, that is why sea urchins are considered as deuterostomes. In fact, the reason we study a lot about sea urchins is that even vertebrates are deuterostomes, and so there are similarities in their development. Now, the mouth fuses with the archenteron to form a continuous digestive tube, and then now once that continuous digestive tube is formed, the digestive organs can start forming. This whole process is actually shown in the black and white figures also, where we can see the extension of the archenteron and the place where the archenteron meets the blastocele. Ultimately, through other kinds of cellular movements and proliferation, the continuous digestive tube is formed and we will have the formation of the digestive organs. In the end, we end up with a larvae that is shown to the extreme left. With this, we come to the end of understanding how cleavage and gastrulation occurs in sea urchins.
Following gastrulation, organogenesis occurs, and we will ultimately form a pluteus larva. The larva that is formed will undergo metamorphosis to form a juvenile sea urchin. The juvenile sea urchin will then grow to give rise to an adult sea urchin. With this, we come to the end of our talk where we learned about the process of gastrulation in sea urchins. We learned about different cell movements and the role of epithelial mesenchymal transition. We also learned about the formation of the archenteron and how it elongates through the process of convergent extension. Finally, we also learned about how the mouth and the anus are formed in sea urchins.